Hello, this is Thomas Warwick, welcoming you to this Bite-Sized Bio webinar, which today is sponsored by Molecular Machines and Industries. Molecular Machines and Industries is a leading provider of microscopic single-cell isolation tools. They specialise in single-cell separation, laser microdissection, and optical tweezers for a wide range of clinical and research-based applications in life sciences, material sciences, and healthcare. Today's presentation is titled Unleashing Fibrotic Secrets, Coupling the Power of Laser Microdissection with Mass Spec, and is being presented by Dr. Jeremy Herrera from the University of Colorado Anschutz Medical Campus. Jeremy Herrera is currently an assistant research professor at the University of Colorado. His goal is to become an expert in the unbiased analysis of proteins, e.g. spatial proteomics, in lung fibrosis. Utilising the power of spatial proteomics, he intends to understand the crosstalk between epithelial cells and fibroblasts to define novel mechanisms of fibroblast activation. As always, we will have a question and answer session after the presentation, so please type any questions that you have into the questions box, which appears on the top right panel of your screen, and I'll put them to Jeremy at the end. Details of how to access the on-demand recording of this webinar will be sent to you by email shortly. So now, over to you, Jeremy, for the presentation.
Thank you so much for that introduction. Today, what I want to talk to you is about laser capture microdissection coupled to mass spectrometry so we can unleash fibrotic secrets. The disease that I research is idiopathic pulmonary fibrosis. It is a very heterogeneous disease. It is characterized by the UIP pattern, usual interstitial pneumonia. And what UIP means is, I always go back to this image here where you see very normal lung. You should see a lot of whole regions where alveolar structures exist. And immediately adjacent to this, you see very dense fibrosis. And there are two hallmark lesions of idiopathic pulmonary fibrosis. First off is the fibroblastic foci that exist within the transition zone of normal and fibrosis. These are sites where fibroblasts accumulate, where they deposit extracellular matrix. Alternatively, honeycomb airways exist in the very dense regions of fibrosis. These are cystic airways that deposit mucus into the lung. So here's an enlarged image of the fibroblastic foci that we have stained with pentachrome. Pentachrome was stained for 5 colors. First off, what you see is this blue color will demark where the fibroblastic foci resides. Adjacent to that, you always see this rich, mature scar tissue that follows it. We theorize that the fibroblastic foci is a fibrotic front, where the fibroblast will migrate invade towards this adjacent alveoli structures. So this is a very heterogeneous disease that really are microscopic lesions. This is a 20x image, and it's really hard to understand how these lesions develop and even progress. Here's another image of the honeycomb airways. This is an Alcium blue periodic acid shift base stain, and blue-purple will stain mucus. You can see here, this is a mucus plug within a honeycomb airway, and to the side is the honeycomb airway cells. You can see the mucus being centrally deposited into this cystic airway. So how do you understand the functions of these lesions in IPF? So what I use is laser capture microdissection to precisely capture regions of interest. So there's a lot of advantages to using laser capture microdissection. It's really, really easy to learn, first off. And you can really capture the areas of interest that you're looking for. And I'll show you some videos of that in the next few slides. Another important thing is that we're using FFPE blocks as a starting material. So these are fixed tissues processed that are sitting on your bench for a year or a decade. I've used really old specimens, and these work just fine for mass spectrometry. And in addition is we process these for a standard H&E stain, hematoxylin and eosin, you really need to see the tissue morphology in order to identify your lesions of interest. So this protocol has been optimized so you can start from an image, capture the region of interest, and do your mass spectrometry. Some of the disadvantages to this technology is, yes, it's a little time-consuming process, and overall, it really requires a pathology-trained user. But once you know what you're looking for, you have the dedicated time. It's just a matter of laser capturing the material you need and processing for mass spectrometry. So what I wanted to show is laser capture in real time. What we're looking at here is an airway from a control specimen. You can see the airway cells here. It's a really structurally intact airway where you see smooth muscle lining the outside, and you can see the adjacent alveoli. And I hit play. And what you're going to see is this line that's following the tissue is the laser that is cutting through to isolate this airway. Again, this is real time, so it goes quite rapidly. And now the cap is going to go on top to lift your tissue of interest. So now you have this specimen. You can record the image, take an image, and then you have that specimen in the tube. So that's a lot of advantages to this technology, so you can always go back to your original images. So the next I'm going to show you is the distal airway in an IPF specimen. So this is an airway that is not in that fibrotic region, but more in that interface that have normal structure, but are actually abnormal. You can see that in this video, we're laser capturing the airway, and you notice there's a bunch of junk that has fallen into the airway. You can see that I'm avoiding it here because I do not want that to end up in my mass spec samples. So now let's talk about the hard stuff. So honeycomb airways have lost their structure. They've lost the smooth muscle cell around their regions of dense fibrosis. And this technology works well to isolate just these little thin airway cells, and I can show you that here. So here we're cutting around carefully on the bottom of the airway cells and then avoiding this mucus plug that's in the middle. And you can draw multiple lines, and here I drew another line and just continue to collect. And now we have our cap that's going to go on top and collect that into a tube, which is quite nice. So what does this look like? So you're going to ask yourself, okay, I've laser captured. How much material do you have in your tube? So I've done, this fun little experiment to show people what this experiment looks like. So here I cut out this honeycomb airway with mucus. You can see that's cut here. And this is a 0.5 milliliter tube. And there's different volumes that you can collect. So you can get up to 0.05 cubic millimeters or 0.01 or 0.001. When I first started this journey of laser capture mass spectrometry, I'm using 0.1 cubic millimeters. So you do pool lesions into a cap or multiple caps from the same patient. But then I started to use different mass spectrometers that are more highly sensitive and getting down to 0.01 and even down to 0.005 cubic millimeters. So we'll discuss that data and the amount of proteins that you can detect using different instruments and different volumes of tissue. So the protocol that I've developed for spatial proteomics is what I call it, is shown here. And you can also go back to my original paper in 2020. So we start off with H&E stained 5-micron section. You cut out your tissue of interest. And when I started this, I am not a mass spec expert. You go down to the books, you read different papers, And laser capture mass spec has been around for some time. And what I did was I copied different protocols and put it together to optimize it for my purposes. So the first step is I add samples with SDS and heat. And what this does is break down the proteins. It helps denature and also undoes the cross-linking that's associated with formalin fixation. The next step is shearing. So we apply this to another technology where you put your tissue sample into a glass container that has a steering rod that sends pulses and pulverizes your tissue. In the end, it looks like little dust that's in your sample, which is important because you want to break your tissue apart as finely as possible to expose the different proteins that are in your sample. The next technology that we used was these S-trap columns. These S-trap columns were designed to absorb your sample in the resin, and it actually reduces the loss of your protein. Other techniques, the mass spec facility said if you do the standard techniques, you're going to lose half of your protein. If you're starting off with such small volumes, it's going to be hard. So the S-trap columns really helped us to minimize the loss. And then we continued down to desalt and perform mass spec. So we did a proof of concept experiment where we laser captured either blood vessels in a 3 IPF specimen. So you can see blood vessels here stained with the pentachrome. This is the H&E. And here you can see these were laser captured and then kept into one tube. And then we also got the adjacent alveoli. These should be very, very different types of specimens. So the expectation is that we will be able to discriminate these regions based on mass spectrometry. So the volumes used for this is 0.08 to 0.09. And the mass spectrometer that we used was the QEHF. In total, we're able to detect 1,200 proteins. If you look at the volcano plot, things that are increased in the alveoli are many, many cytokeratins, which is consistent what we should see in the alveoli. Increase in the blood vessels, highlight a lot of these proteins here, are proteins consistent with blood vessels. So we are on the right track. We also did a PCA analysis and showed that the samples separated by region, so the blue is alveoli and the red is blood vessels, and not by patient. The patients are 1, patient 2, and patient 3. So this was quite exciting that they separate by region. And the next thing we did was we wanted to test the sensitivity. So in this experiment, we used different volumes using the same protocol, same instrument, and show that if we get down to 0.006 cubic millimeters when we start to taper off on peptide counts. So this was great for 2020, but we can do better, and I'll show you that in the following slides. However, when we were working on publishing this paper, I was already laser capturing to understand what's the function of the IPF fibroblastic foci. So again, the fibroblastic foci is a hallmark lesion of IPF. So you can see that we can capture the fibroblastic foci. That all goes into one tube. On the same slide, I went back and laser captured the mature scar tissue. Great, so I didn't need more slides. And the same specimen, we go back and now we capture the alveoli. So we're really getting to the spatial orientation of this disease, resolving that heterogeneity. So this experiment used an N of 6 IPF, N of 6 non-fibrotic controls. And for this volumes, I used 0.1, which was consistent with the previous alveoli and blood vessel paper, using the same instrument, QEHF. So first off, on the left, I have a Venn diagram showing that we detect 3,000 proteins. I was really excited that we were able to detect 3,000 proteins because, again, this is fixed and stained tissue. I know in the previous slide, we detected like 1,200 proteins, but one thing you have to understand is that alveoli are going to have a different protein makeup than fibroblasts. And we're also going to see different types of proteins in mature scar tissue. So the heterogeneity between different regions of your tissue, you're going to detect proteins that are uniquely expressed there. So the more variety, the more proteins we're going to detect. And also the more samples that you have, the end samples, the more proteins the mass spec is going to start to detect as well. When we look at the PCA analysis, I got really excited because the non-fibrotic alveoli separate from the IPF specimens as the first visualization. However, the most distant is the non-fibrotic alveoli to the fibroblastic foci. The IPF alveoli cluster closer to the non-fibrotic alveoli, and the mature scar tissue is in this green color here, which is suggestive that, this could be a trajectory of fibrosis progression. So it's quite nice that we see this type of separation. So we did bioinformatics. You can create a lot of interesting charts with this type of data. Looking at the volcano plot, we're able to detect about 300 proteins that are changing between these regions. If you do halfway analysis, the themes that are upregulated in the fibroblastic focus are as extracellular matrix, collagen biosynthesis, and everything associated with extracellular matrix. This affirms what pathologists know, people in our field know, that the fibroblastic foci is indeed the site of collagen biosynthesis. And now this is proven using spatial proteomics. When we look at the extracellular matrix proteins, if we compare the IPF fibroblastic foci to the left, non-fibrotic alveoli to the right, and matrix proteins broken down to the different categories, you see a complete shift like collagens. Collagen 4s are basement membrane proteins, which are associated with more normal lung tissue, and blue are increased in the alveoli versus decrease in the IPF fibroblastic foci. More other collagens that are associated with fibrosis, like collagen 1, collagen 12, are increased in the fibroblastic foci, decrease in the non-fibrotic alveoli. So we're really understanding the composition of the fibroblastic foci as well, which will be important if we're going to build 3D models. And then everything else follows suit. So it's a very powerful technology to define different regional areas of the fibrotic lung. And if you want more information, you can go to my paper published in 2022. So what about the honeycomb airway? So we also laser captured not just the honeycomb airways, but the mucus plug. So for this strategy, I cut the mucus plug first because I don't want that to accidentally fall into your honeycomb cyst airway specimens. So I cut out the mucus plugs, and it makes it easy because then when you go back to your tissue, you know where the hole is, and then you can find where the honeycomb cyst is, and then you can laser capture that into separate tubes. So for this experiment, we used 10 fibrotic specimens, 6 controls, and this got a little bit trickier because these are so thin that, to save myself time, I'm like, okay, let's use 3 times less volume. We use 0.03 cubic millimeters. But at that time, the mass spec facility said, hey, Jeremy, we got this Orbitrap Explorer that's supposed to be 10 times more sensitive than the QE-HF that we used before, so we just gave it a go, right? Just wish for the best. In total, for this experiment, using 3 times less volume, detected 2,600 proteins. which is a good sample size. But when we compare the fibrotic distal or uninvolved airway cells to the fibrotic honeycomb airways, what we see is a lot of proteins associated with mucus biology are increased. MUC5B, which is one of the main mucin-forming proteins, and BPIFB1 is also another protein that's always associated with mucus. AGR2 is another protein that is essential for mucus biosynthesis. So we confirmed that these honeycomb airway cells are the site of mucus biogenesis. And you can follow this work in Respiratory Research 2023. Right. So this is the work that I did in the University of Manchester, UK during my postdoc work. And I was recruited to the University of Colorado, working under the mentorship of David Schwartz, where we are interested in understanding how the gain-of-function MUC5B promoter variant, which is the dominant risk factor for IPF development, impacts the development of fibrosis. So the question that we want to address is, does the MUC5B promoter variant have a different proteomic signature? And the hypothesis that we have is that when you have a normal lung that has the MUC5B promoter variant, is that these lungs are vulnerable to a second impact that would drive IPF development, whereas patients with the wild type allele, although they do develop fibrosis independent of MUC5B, we might learn a lot about the proteomic signatures that can be drivers of lung fibrosis. So for this strategy, we used 12 controls, 12 IPF, 6 with the promoter variant, 6 wild type in both cases. We again interrogate the honeycomb airways, looking at normal airways to those normal distal airways in the fibrotic lung and also the honeycomb airway. We also interrogate the fibroblastic foci, the same experiment, alveoli from the fibrotic patients as well. And we also this time look at the transitional cells, and I'll explain what the transitional cells are in the next slide, and also the fibroblastic foci. So transitional cells are this, not a new cell type, but a recently characterized cell type that is of interest to our field. They are located right above the fibroblastic foci. So here's an H&E of a fibroblastic foci that is characterized by the type 1 collagen expression. So you can see the collagen kind of shoots up more towards the normal alveoli, consistent with the idea, and you can see lower here, that they are progressing towards the alveoli. And the transitional cells have been shown to express cytokeratin 17, so we can really demark them here. So what we've done was ask the question, can we laser capture these transitional cells? This was quite a challenge because they're really, really small. So when I did this experiment, you can clearly see that we can capture this. I used a volume of 0.005 cubic millimeter. This was not because I wanted to do this small volume. This is just because this is how much I can isolate from the IPF lung. And we went with that. I was quite happy with 0.005. And this time in Colorado, we're using the timsTOF SCP. SCP stands for single cell proteomic system. So this is, top of the line mass spec instrument with single cell capacity. So we wanted to test how many proteins we can detect with this volume. And the answer was 4,900 proteins, which, really, really impressive. However, we're working on modifying my protocol to make this better. So let's look at the data. We're looking at a heat map analysis based on the top 500 variable genes, where we have control airway, IPF distal airway, IPF honeycomb airway. we have the ingrained IPF fibroblastic foci, and then control IPF alveoli in the transition cells. Very impressively, all the airway samples, whether you're control or IPF, cluster in this region. The fibroblastic foci cluster on their own, right smack in the middle, the most different from the airway and the alveoli. And the third cluster is the alveoli and the IPF transitional cells align themselves more with the alveoli and are the most distant from the airway cells. And I know this is an important experiment because we're not quite sure, what the biology of these transitional cells are. There's a few papers, and it's just an emerging theme in our field, but this is quite exciting. So what I want to now focus on is these transitional cells. Looking at the Venn diagram, we detect about 2,000 proteins, and with 380 proteins uniquely identified in the IPF transitional cells. When we do pathway analysis on this gene list, we see things consistent with what other people are publishing. These are really stressed cells. We see cellular response to stress as the top pathway, and apoptosis is also on the list. So what these cells are doing over a fibroblastic foci that we know is the only site where collagen biosynthesis occurs, there could be some cell communication with fibroblast. If we look at the volcano plots, we detect way too many proteins. We detect like 600 protein changes. When we do pathway analysis and things that are increased in the IPF transitional cells, we see things consistent with eukaryotic translation initiation and elongation. And downregulated is mTOR signaling pathway, which impacts translational control. So not only are these cells stressed, but they are active in translational control, which are themes of lung fibrosis. So let's talk about the MUC5B promoter variant. So this was the main aim of this project, was to identify which proteins are MUC5B driven. When we look at IPF specimens, where we had the MUC5B promoter variant to the right, wild type allele to the left. And none of these proteins meet our threshold. Whether you're looking at the IPF alveoli, IPF distal airway, or IPF honeycomb airway, it flatlined. However, the exciting thing is that when we look at the non-fibrotic control specimens, again, these are patients without fibrosis. We start seeing early changes. We detect about 100 proteins that are changing in the alveoli. Again, MUC5B is a gain-of-function promoter variant, and it's shown to be misexpressed in both alveoli and airway cells. And here we're showing that it's potentially impacting the alveoli to a greater extent. We look at the non-fibrotic control airways, we see two proteins that are increased in a wild type allele. So this is quite exciting data. Actually have a first draft of this manuscript in preparation by this week. And hopefully by this summer, everyone will be able to read our findings on how MUC5B is driving pulmonary fibrosis. So in conclusion, what I want to state is that laser capture coupled with mass spectrometry is a powerful technique that will overcome tissue heterogeneity. I've applied this technique on mouse samples. That works just fine. I have other colleagues using fresh frozen tissue, and that also works too. So there's a lot that you can do. It doesn't have to be formalin fixed, paraffin embedded. It doesn't have to be human. It doesn't have to be, mouse. It doesn't have to be a lung. It can be whatever you want. So long as you can see your tissue specimen or region of interest, this technique will work. It also works incredibly well at low volumes, granted that you're using the single cell proteomic system. And if you're not, maybe you have to go back and laser capture some more. And, in this talk, I've defined the molecular functions of the IPF hallmark lesions, the fibroblastic foci, the honeycomb airways, and now the transitional cells using this technology. And also, we show that the MUC5B promoter variant impacts the proteome of control specimens, not when fibrosis is established. And with that, I would like to acknowledge, our very large team with Professor David Schwartz and Ivana Young, and my collaborators, present at the proteomics facility here who have helped me along the way with the single cell proteomic system, the histology facility, specifically Professor Ruben Tudor, who owns one of the MMI instruments, which works wonderfully. My previous advisors at the University of Manchester and others back from my PhD days. And I thank you, and we'll take any questions that people have.
Thanks, Jeremy. That was an excellent presentation. You clearly get amazingly rich data. Several thousand proteins is extremely rich. It's almost too much in some instances to drill down into, and it wasn't so long ago that protein mass spec was incredibly hard. So we've got a few questions from the audience, and please feel free to post questions that you have into the questions box on your right. So is it possible with your cell cut laser microdissection system to visually inspect the sample that you've excised? And how does that process work?
Yes, that's a great question. So using a laser capture instrument, you can go back to your tissue, go back to the cap and visualize what's on your cap to see, did you capture the right thing? And that is definitely doable. So you can take images of the captured sample as well.
Cool. Do you recommend that as a step before going to mass spec to always double check what you've excised?
I would do that initially. Once you become more comfortable with the technology, you'll start not to do that.
Yeah.
Once you get to so many samples,
It's not doing it in your sleep. So can you share information regarding the time needed to isolate the sample of interest? I don't think you touched upon that in your talk.
No, so that's really dependent on the instrument that you want to use. It's also dependent on what you're cutting out. So if you're cutting out fibroblastic foci, maybe give yourself 30 minutes. Those transitional cells or honeycomb cyst airways, which are really small and thin, I would say maybe 2 hours per patient. Yeah. The alveoli is quick. I mean, if you have nice alveolar structures, it can take you like 15 minutes to get enough volume.
Yeah, I guess it's really crucial to make sure you only get the sample of interest, so you don't get mass spec results for loads of noise essentially. So someone's asked if you tried your mass spec protocol with OCT embedded tissues rather than FFPE, and would the protocol work?
I have not personally tried OCT, but I have colleagues who have done this successfully. I believe they used human brain.
Okay.
So either brain or breast tissue, and they're able to yield results. So that's fine.
Cool, thank you. Have you ever found that the H&E staining reagents have interfered with your mass spec, or has that never been an issue?
This is a really great question. So it's been shown that I believe hematoxylin does negatively impact protein detection. However, I still include it because it's easier to see what I'm cutting out this way, and I just take the blow through protein detection for finally capturing what you're interested in. I've not tried other stains like laser capturing an LCM blue stain or a pentachrome stain. I imagine that'll work, or if you wanted to do an antibody detection, so use IHC and then laser capture that. I'm sure it's going to work, but then you might interfere with detecting the antibody as well.
Sure.
So there's different, yeah, I've not tried it. I know it'll work, but there will be some difficulties along the way.
Cool. We've got lots of questions coming in. Let me just work through these. So someone's asked, do you measure the tissue itself or the membrane cut volume? I don't know if that means anything in
Can
particular
you
to
repeat
you.
that question?
Do you measure the tissue itself or the membrane cut volume? I don't know if that means anything to you. Maybe you're not
No.
quite clear what they mean. I'm not sure either.
Yeah.
No worries. So I had a few questions as well. So did you get good coverage for membrane proteins from your data? Have you looked into that? Or is it typically extracellular, soluble proteins?
So yes, I usually go for the soluble proteins. Currently, we're isolating the insoluble fraction, and we're actually detecting a lot of proteins in that too. So we're interested in looking at matrix cross-linking effects and looking at what else is there. So, but yeah, we detect a wide range of proteins. It's not just all like matrix proteins. And
Sure.
if you're
Okay.
interested in a specific protein, if you know the character of your protein, you can change the protocol to add the buffers that will solubilize it better.
Nice. So someone's asked, just following up on the issue of hemataxylin, are you aware of how the hemataxylin reagents interfere with mass spec, or do you just know that it does?
I just know that it does. I've read it in papers that it reduces the protein yield. Um,
Sure.
so yeah, I haven't looked into that.
No worries. So how many cell or tissue samples are needed for sufficient quality of the mass spec analysis?
That's a really great question. It really depends on what type of mass spec instrument your facility has. So if you have the older versions, you're going to need more volume. I never really count cells because I just look at volume as the final readout. What's the volume of tissue that you're using? Because we're not just capturing cells, you're capturing the matrix that the cells are embedded in as well. So it's not just cells.
Cool. Okay, thank you. And somebody's asked, how do you lyse the collected material in the sticky cap they're collected in? I guess they mean
Yeah,
process,
yeah.
but
I actually get asked this a lot, and I should have wrote it in the paper. So if that you have this cap, the sticky cap where your tissue is sat on. So initially, I try to like pry things out, and I've seen people remove the entire jelly and put that in the
yeah,
tube. Don't do that. What I do is I get a 200 microliter tip, and I snip it sideways, like at a 45-degree angle, and then I dip that into your buffer solution. And when it's a little wet, then you just scrape the tissue onto that tip, and the tissue sticks there, and then you just put it back into your tube, swirl it around, and your tissue would just fall off. So that's the way forward. Don't try to centrifuge it. Don't try to like mix your solution up and down with your tube because it's not going to fall off that way. So scrape it
sure.
in.
Neat. So I guess you could take these two questions together. So someone's asked, what microscope was used for this study, and someone else asked, what's the greatest thickness that you could cut with this
Yeah.
microdissector to increase the tissue volume?
Yes. So I've gone up to 10 microns to put on a laser capture slide. You can go thicker and it'll cut like 30 microns, but the reason why I don't do that is because you're not going to be able to see the morphology of your tissue once you get more than 10 microns. So those pretty images that you see are 5, and I usually go to 8 microns afterwards and just adjust and then laser capture that without a problem. And what was the type of microscope? I don't know, whatever the MMI setup is.
Sure, thank you. No worries. I think we can share that one after the talk. So I was wondering, you mentioned all these high numbers of proteins you detect. What is the criteria, or do you have criteria that you use to determine whether or not you successfully detected a protein? Do you look for a certain number of peptide matches to a target sequence?
Yeah, so that's all done with the bioinformatics software. It depends on how many peptides were identified for that specific protein.
Yeah,
And once that threshold is established, and if it was detected in your sample, some samples it won't detect it, but in other samples it will. So
Sure.
the criterion that I usually use now for the manuscript that's in preparation, if it's detected in 80% of those samples, then we say it's detected. So
Okay.
that's a loose criterion, and everyone has different criterions, which is
Yeah.
their
One of
manuscript.
those things you set once you've got your data.
Yeah.
So someone's asking here, or they're just asking about the size of the capture and the error rate. I presume they mean error rate in detecting, proteins. Have you noticed any correlation between the size of your capture and the confidence you have with protein detection?
I've not I've not specifically tested that experiment. I know we've tried different volumes of tissue to see how many proteins were identified, but I didn't chase specific proteins to see was it
Yeah,
detected only in this volume versus the other ones. Um, the good thing about this technology is that you're really capturing your region of interest. So it's
yeah,
already an enriched pool. So, the data that I get so far is pretty consistent with the biology of the cells.
yeah. It certainly looks that way from your talk. So someone said in their experience, sometimes the tissue can get in between the membrane slide and the glass slide. Do you have any tips for collecting tissue on the sticky cap? I guess you touched upon this with your previous
Yeah,
answer.
yeah, yeah. So, yes, it's true. When you laser capture, you're getting that membrane from the MMI slide. That's just going to have to go into all the samples. So that actually is lost when you pass your samples through the S-trap column. So I noticed that the little membrane plastic is stuck there. But it also depends on the detergents you're using. I know initially the mass spec wanted to try their protocol, and their reagents pretty much chewed apart that plastic. It was not good news for their
Okay.
mass spec instrument. So you don't want your conditions harsh enough where that gets dissolved, because then the mass spec facility won't be happy with you.
Yeah, they're not cheap instruments.
Yeah.
Um, so with the emergence of spatial transcriptomics, does this imply laser microdissection will no longer be necessary in the future?
Uh, so I know other people have done laser capture transcriptomics. I know you guys had a talk on this. Was it Lavinia? We gave a talk on that recently. But spatial proteomics has its own niche and need in the field. I mean, there's a lot of platforms out there that you can buy, like 10x, NanoString, to do spatial transcriptomics. But we know that proteins are the functional units of a cell wall,
Yeah,
mostly
yeah.
functional units. So there's definitely still a need for laser capture, unless this can become automated or when technology gets better. Uh, yeah, but
I guess it's
currently
a
there's
case of,
no platform you can just purchase from 10x NanoString to do mass spectrometry spatially.
I guess it's that classic thing of do both, right? If you can afford it, why choose one technique when you can approach your research question with two? Um, so that's the end of the audience questions. I guess a nice one to wrap up with then was, where do you see this technology developing from here? Is it all about detecting more and more proteins from a given sample size, or are you interested in coupling this with native mass spec?
Yeah, so I would like to build upon this by doing either phosphoproteomics. We're also going to start doing other post-translational modifications as well, to see what else we can learn from laser capture mass spec.
Okay, well, thank you. So that's the end of the questions that have come in, and that's all of my questions. So I guess that brings us to the end of the webinar. So thanks again, Jeremy. That was a really great presentation. I certainly enjoyed going back to my protein roots, and thanks also to our sponsor, Molecular Machines and Industries. And finally, thanks to you, the audience, for taking the time to attend and listen in today. So until next time, good luck in your research, and goodbye from all of us at Bite Size Bio and Molecular Machines and Industries. Thank you.